thank you very much. Thanks to all of you for coming today, and thanks for DNA Genotek for providing the opportunity to present today. First of all, my disclosures, primarily publicly funded granting agencies, typical for sort of an academic research lab, but also I think most importantly that one is thanks again to DNA Genotek for providing the opportunity and supporting travel here to this meeting, but also I think you'll be interested to know some of the data that we're presenting today, that I'm presenting today, is based on a year ago DNA Genotek had their first open grant competition. And we were fortunate enough to get some funding to do some of this research. And so we had support from them for that. So first of all, I just wanted to present this slide really by way of sort of orienting you to the work of our lab and to provide a bit of a caveat and an acknowledgement. So first of all, our lab is really focused on looking at the neurobiology and genetics of OCD, and we're looking at that at multiple levels of analysis. The caveat is that really I'm relatively new myself to the field of epigenetics. Most of my work has been focused at the area of genetics and at the level of sort of phenotype. We've done a little bit of work recently in intermediate phenotypes such as neuroimaging. The work on epigenetics, I really want to acknowledge Dr. Rosanna Wexberg. So a lot of this work has been collaboration between our lab and hers. Dr. Wexberg is also at the Hospital for Sick Children, as well as a postdoc, Dr. Darcy Butcher, who's done a lot of the work on this project. So by way of background, Obsessive-Compulsive Disorder is a neuropsychiatric condition characterized by persistent intrusive thoughts or obsessions and repetitive ritualistic behaviors known as compulsions. It's a common disorder, so about 1 to 3% of individuals will develop OCD, clinically significant OCD, in their lifetime. It's been listed as one of the 10 leading causes of disability according to the World Health Organization, and that's sort of across the board, all medical conditions, as well as psychiatric conditions. And OCD we know is a phenotypically heterogeneous disorder, so it's heterogeneous with regards to differences in age of onset, differences between males and females, differences in symptoms, etc. And we think that this phenotypic heterogeneity may reflect underlying genetic and neurobiological heterogeneity. Some highlights of the genetics of OCD: we know that it's heritable, the heritability being up to 65%, with that heritability being increased when twin studies are done on samples based on child probands. Most of the remainder of that variance is non-shared environment. We know that there have been more than 80 candidate gene studies of OCD, which have been reported with mixed findings, again, typical of other complex disorders. One of the most consistently reported findings was for a gene known as SLC1A1, glutamate transporter, from our group. However, it's been difficult to nail down which specific variant or haplotype within SLC1A1 is implicated based on meta-analysis. There has been one genome-wide association study, which we were involved in. Our group was one of the leads on that, and it revealed some interesting candidate genes, including genes involved in postsynaptic glutamate transmission. However, again, as typical with complex neuropsychiatric disorders, none of the findings was genome-wide significant, suggesting both that there's some significant missing heritability, but also that larger sample sizes may be needed. So why epigenetics? One of the reasons for that kind of follows on what I was talking about, in that much of the genetic variance remains unexplained. And epigenetics is one of a number of mechanisms that have been proposed to account for this missing heritability. But a second important point is that actually in some ways similar to we were hearing about telomere length, we know that DNA methylation and some other epigenetic mechanisms are known to be influenced by the environment, so environmental stressors of various sorts. And that fits well with what we know of OCD, for example, because twin studies suggest, as I mentioned, that much of the variance is environmental in nature, and also that we know that OCD symptoms wax and wane in response to a variety of biological and psychosocial stressors. The mechanisms for those, though, are unexplained, and it's perhaps epigenetics that may explain that link. So what is epigenetics? Again, I know this will be review for many of you, but just in case you're not familiar, what we really mean by epigenetic is that a heritable change in genome function that occurs without a change in the nucleotide sequence within the DNA itself. And there are a number of epigenetic mechanisms. The one that I'm going to be focusing on, and where we've focused much of our work, is DNA methylation. We know that DNA methylation and other epigenetic mechanisms have been implicated in a number of neurodevelopmental syndromes. In particular, though, we focused on methylation. Again, this will be familiar to many of you, but as a review, DNA methylation occurs at the Cs of CpG sites. CpG sites are distributed non-randomly in the genome. And in fact, most of the action in epigenetic studies has been on what are known as CpG islands, which are areas that are from 200 base pairs to several kilobases in length, comprising 1 to 2% of the genome, which have a 5-fold greater frequency of these CpG dinucleotides compared with the rest of the genome. We do know that methylation of promoters containing CpG islands can cause gene silencing, so it can have an obvious functional effect on expression. Again, a reason for the interest in these areas. And that DNA methylation patterns are established by a number of enzymes in the de novo methyltransferase family. DNA methylation is responsible and required for a number of normal processes, including DNA imprinting, X chromosome inactivation, and I think actually of interest and something to always keep in mind, tissue-specific gene regulation. And that particular issue causes something that has to be kept in mind with research, because whatever tissue type you're looking at, it's possible that the methylation changes you're seeing are tissue-specific. Although, as I'll demonstrate to you, I think we can see that some of these changes are quite widespread across different tissues. We know that aberrant DNA methylation may be implicated in a number of neuropsychiatric disorders. In mouse models, epigenetic alterations in the brain have been associated with social and attachment behaviors and in various forms of learning, including fear conditioning. An altered DNA methylation has been found in human studies, in blood and in brain tissue, postmortem brain tissue, of patients with a variety of conditions. Just to name a few: autism, spectrum disorders, schizophrenia, major mood disorders, etc. There's also been some interesting findings of relevance to psychiatric phenotypes of major DNA methylation differences based on history of early adverse childhood experiences such as child abuse. So to talk about the work that we've done, I'm going to be presenting work from 3 linked projects. The first is one that we did, the first project we did prior to the funding we received for this from DNA Genotek. This was based on blood from a very small sample of 10 OCD patients and 16 controls, tested using the Illumina 27K array. The other 2 studies are based on data from a newer array, the Illumina 450K array, and as the name implies, obviously many more probes, 450,000 versus approximately 27,000. We looked in our second project, we were interested in intra-individual variability. So we were looking at 12 OCD patients in whom we had both saliva and blood, and we were comparing the saliva and blood for differences in methylation. And the third project that we did was looking at 30 OCD patients and 30 controls using, the Illumina 450 array. So even though the array types are different, it's important to note that they follow similar basic principles based on the sodium bisulfite conversion. So the first study, in which we looked at pediatric OCD, and this was in blood, again, 10 OCD patients, 16 controls. Average age, by the way, is about 11, and these are patients that have been selected for being medication naive. So we're trying to eliminate that confound as much as possible. We did this analysis using the 27K array. It's important to point out that this array is really very focused on the CpG islands that I mentioned that are primarily in promoter regions. So they really put emphasis on that, whereas there's broader coverage in the newer array. A lot of the data I'm going to be showing you uses what are known as beta values, and these are really levels of methylation. It's a ratio of methylated cytosines within these CpG islands versus methylated and non-methylated cytosines. And you get a value between 0, which is unmethylated, completely unmethylated, to 1, which is fully methylated. Sometimes when we talk about differences, we talk about a percent difference between our cases and controls. But again, that's based on these beta values. In this initial study, we looked at we set out thinking that we weren't going to find necessarily epigenome-wide significance because we had such a small sample size, but we did want to be fairly conservative. So we looked at least nominal significance, and then we wanted a conservative cutoff in terms of large effects, and we chose an effect size of 17%. Now, as you'll see, the effect size are actually quite large, and some of these would have actually survived multiple comparison corrections despite the small sample. We identified 40 CPG sites with loss of methylation using this method, and 14 CPG sites with gain of methylation in blood samples of OCD cases compared to controls. And I'm going to show some examples, and I think in all the data I'm showing you, it's not necessarily because these are still, again, relatively small samples. I think more work needs to be done. It's more just to highlight the feasibility of these methods and also to show you that many of the findings do fall into interesting pathways that have been implicated previously in OCD, which we were quite pleased to see. So first of all, we identified a loss of methylation in the promoter of IL-16, interleukin-16. So here we have OCD cases, loss of methylation versus controls. There's quite a clear split between them. You will see actually in some of these slides that there's a fair bit of variability often in the patient groups, and we think that could be due to a variety of reasons, which I'll comment on. But in this case, there's a pretty clear split, and we were interested in this particular candidate for this particular finding for two reasons. One is that this IL-16 does have a neuronal-specific isoform, which has been shown to co-precipitate with glutamate receptors, particularly NMDA glutamate receptors. And our group and others have actually found genetic associations with NMDA receptors, and we found those to be associated with neuroimaging phenotypes of interest. There is also a hypothesis that and evidence that there may be some children with OCD in whom the symptoms are triggered by an autoimmune phenomenon. Basically, they're post-infectious in nature. So again, having an interleukin gene, a cytokine gene involved was of interest to us. Second finding was gain of methylation in the promoter of ABCA2. Here you see a bigger range within the patient group, but it's still higher, significantly higher methylation. And in this case, we found it of interest because this particular gene is involved in myelination. Again, of importance for any brain disorder, but with OCD, there have been white matter changes observed in imaging. And finally, with this first study, we found loss of methylation in the promoter of GRIM2, and this is a metabotropic glutamate receptor, again of interest to us because of the evidence for glutamate signaling changes in OCD in general. So moving on to the second two studies, these were based on the methylation 450 array, Illumina methylation 450 array. And to remind you that this array has broader probe coverage across the whole genome. It includes, for example, areas known as CPG shores. So the initial expression CPG island is sort of extended to these areas that are a little bit more outside of the gene and outside of the promoter that are enriched in CPGs. And this particular array, as the name suggests, covers more than 450,000 CPG sites. One small study that we did using this array was to look at blood and saliva from within the same sample. I think I may have said on the previous slide they were patients. In fact, these are actually controls. So we're interested in just looking at a control sample in whom we had both blood and saliva collected on the same individuals. And we did identify DNA methylation differences in a small number of probes. But actually, that was a relatively small number, which was of interest to us. That significant, large differences we really only found in about 4.7% of the probes. And again, suggesting that there may be some similarities and more similarities than one might expect. Or perhaps that one might expect to some extent, because as was mentioned in the previous talk, we know that much of the DNA in saliva is derived from white blood cells. We're interested in looking more in depth at the different tissue types and how that might explain the differences we did find. This is just an example of where we did find a large difference and one that wasn't, when you look at the gene, not that surprising. So this is BIN2, bridging integrator 2. It's a gene that's expressed in hematopoietic tissues. It may be different because of different cell types between saliva and blood. So finally, to talk a bit more about our more extensive saliva study of 30 pediatric OCD patients and approximately 30 controls, we ended up with 29 controls after quality control. Again, the age was fairly similar to the other study I showed you. I think it was just slightly older, average age of about 13, approximately 50% male, female, by the way, in both studies. All the samples, by the way, also have had, they're medication naive. They've also had MRI, magnetic resonance imaging studies as well, so we're interested in looking at that in future. Part of the reason we selected these particular samples was because of having that rich data. So we found differences in about 8,585 probes after correction for multiple testing, and we narrowed down on a number of CPG sites in which we found significant difference. They were in these CPG sites of a relatively large effect. Important to point out that there were no significant global DNA methylation differences between OCD cases and age and sex match control. So the differences are really based on these specific sites. So I'm going to give you two examples of what we found, of interesting genes that we found. We didn't find the same genes, that they did fall in somewhat similar pathways. In particular, in one case, this particular gene was involved in loss of, this is VTRNA21. This was a loss of methylation. Now here, two things to point out. Yes, there is a difference between cases and controls, but something to point out that there's a big variability in this case. We actually think it could be as much, yes, there could be some tissue variability with the saliva that we use, but also we think this particular sample may have been a little bit more heterogeneous clinically, and we're trying to drill down on that a little bit. But this particular gene is involved in neuronal migration, which is thought to be implicated in OCD, a neurodevelopmental disorder. We found a loss of methylation in exon 1 of KCNC2. This is a potassium-gated channel, and this falls into the same, I talked about the glutamate pathway. This is a gene that actually its expression is controlled by glutamate, and it's expressed in neocortical neurons, GABAergic neocortical neurons, and it's been implicated in mouse models of some other neuropsychiatric disorders. So I wanted to finish with some take-home points. I think, first of all, we've demonstrated feasibility of obtaining high-quality DNA methylation data from saliva using arrays, and that was one of our major goals in all of this. Like many other groups, we found that collecting saliva is particularly convenient for us. Many of our kids, they don't need to give blood for clinical reasons, and we found that it was a barrier to recruitment when we were only able to collect blood, and it's been helpful. We've also been able to, for example, collect from general population samples using saliva on quite a large scale, and that wouldn't have been possible using blood. So we had an interest in terms of our own research program of being able to do this in saliva. Second point is that we're able to identify significant case control differences, but we've had some interesting findings. We did find a lot of variance in saliva. Now, interestingly, that small study that I showed you of the 12 individuals in whom we had saliva and blood suggests that a lot of that heterogeneity is actually more on an inter-individual, between individual variation rather than intra, although some of it is probably due to heterogeneity of cell types within saliva. I think the strength of our approach is that if our results are confirmed in larger samples, again, why do this? We've potentially identified biomarkers that can be easily collected on a large number of subjects using an accessible tissue. And one of our reasons of interest for that is that a limitation of our approach, something that always comes up, for example, in grant reviews for us, is that, well, what about tissue specificity? You're not looking at brain. And it's a legitimate concern which has to be addressed, and it's being controversial within the field. But I think our group and others have taken the stance that, yes, there is tissue specificity, but there are also a lot of changes, methylation changes that can be observed across tissues. And also, if we're going to look at this as a potential biomarker, we're obviously not going to be able to get brain, and in terms of looking at a biomarker, and therefore there's some value in doing this. And also, as I mentioned, our concern is mitigated somewhat by our preliminary blood-saliva comparison. So you show some changes that most changes are true across these tissues. And we're doing some other work looking at brain and saliva in animal models and looking at how these genes are expressed, our findings in postmortem brain samples. So future directions increase our sample size. Ultimately, we do want to validate these findings, our top findings, using pyrosequencing in a much larger sample size, but we're actually going to somewhat increase we'd like to increase a little bit our current sample size for methylation discovery as well. We're going to examine, as I alluded to earlier, the influence of methylation on neuroimaging measures of brain structure, function, and chemistry. That may also give us a bit of a handle on this heterogeneity. So, maybe there's some underlying intermediate phenotype that they're influencing. And then looking more closely at the relation of specific symptom subtypes and methylation. So I'd just like to end with some further acknowledgments, and thank you very much for your attention.